So I'm going to first start off by saying my job is going to be a lot easier than Rebecca, who had to basically give an overview of a 200-year organization with all its activities and interests. And I'm going to focus in on one particular topic, and it's a particulate matter. And specifically, what are the current USP standards related to particulate matter? So my job today is going to be a lot easier than Rebecca's. I know it's extremely difficult for someone to give a brief overview of a 200-year company or organization with all its activities and efforts. My job is going to be more precise and more focused, and that I'm going to focus in on the topic of particulate matter, and specifically the current USP standards related to particulate matter. Probably the first question is, what is particulate matter and why is it's important? Well, it's important because particulate matter is a critical quality attribute for all injectable drug products. The historical definition, which is the definition used by the USP, the Japanese Pharmacopeia, as well as the European Pharmacopeia, is extraneous, mobile, undissolved particles that are unintentionally present in solution. And so again, when we think about an injectable drug product and this idea of unintentional materials, again, one can start to think about why it's a critical quality attribute, because it can impact not just the quality of the drug product, but the safety of the drug product. When we think about particulate matter within the USP specifically, we kind of divide them into two buckets. We talk about visible particles and we talk about subvisible particles. When we think about visible particles, this is around 100 microns and larger. And I'll say this right now, there's a lot of debate around where the visible range starts and where it actually ends. But again, for the compendial purposes, we start to say you can visually see a particle at around 100 microns. When we think about subvisible particles, it's going to be those particles that are 50 microns and less. So when we think about particulate matter, I like to say that not all particulate matter is the same. And so within the USP, we actually kind of classify it into three different buckets. The first bucket are particles that we consider inherent. And these are going to be things that we expect to see in the drug product. They're things that are natural characteristic of the product. And examples could be microparticles or aggregates associated with biologics, or suspensions, which are particles in themselves. And then we have intrinsic particles. Again, these are going to be materials that find their way into the drug product within the manufacturing space. So it could be a particle that's emanating from the product formulation, from the manufacturing assembly, like a stainless steel vat, they may give off metal particles, or from the packaging system when you during the assembly process where you may have glass particles or elastomeric fragments. And so when we think about intrinsic particles, again, there's going to be packaging material, things related to solution formulation components. It can be even product packaging interaction that leads to particle formation, or again, process-generated particles. And then the final bucket of classification that we have within the USP is extrinsic particles. And these are the ones that, again, out of all three are the ones you definitely do not want, because these are materials that come from outside your manufacturing process. And so you think about dust, paint chips, rust, insect, hair, organic material, and fibers. And so this, again, if we start thinking about a quality attribute for a drug product, safe, again, paint chips is not one of them. So when we think about particulate matter, we have to think about what type of particle it is and what's the potential impact will actually have on the drug product quality, as well as on the potential safety to the patient. Not all particles are the same. So now I'm going to transition into our standards as it relates to visible particulate matters. And again, the idea, again, that we know what particulate matter actually is. And so now how does one go about looking to see if they have it, especially within the visible particle range? Well, anyone who comes and asks me the question of compendial standards related to particulate matter in general, I always tell them the most important chapter is General Chapter 1. And they always scratch their head because they say, well, not 788, not 790. No, I will always say General Chapter 1, because this is the main chapter that talks about the various product quality tests for injections and implanted drug products. We did a major revision to the chapter back in 2015, became official 2016, and then had subsequent revisions 2018, 19, and 20 to make sure the chapter were better aligned with our monographs. If you look in the chapter, it pretty much outlines general quality requirements for the various injectable dosage forms, solutions, suspensions, all the way to drug-eluting stents. And within this chapter, there's a specific section that calls out foreign and particulate matter, which is one of the most valuable quality attributes for an injectable drug product. And within this section that deals with foreign and particulate matter, it kind of outlines some very basic expectations when it comes to particulate matter. The first one is that, again, a drug product manufacturer should do everything in their power to exclude particulate matter from their drug product. Also, we know it's a lot easier to prevent it from being in your drug product than from remove it. And so in the discussion, we talked about particles emanating from within the process. Again, understanding the particle load coming from your packaging components, understanding the particle load coming from your manufacturing line, all these things need to be considered in order to ensure that the particle load that finds its way into the final drug product is minimized. Within this section, it also talks about each final container should be inspected for observable foreign and particulate matter. And again, if any container is observed to have visible particulates, they must be rejected. And so when we look at the lot level, it goes and states that every lot should be essentially free from visible particulates. And I have to say, when I first started working at USP and I became the owner of this chapter, the main question that I would always get in regards to general chapter 1 is, what do you mean by essentially free? Can I have one particle? Can it be a translucent particle? And at the time, the USP did not have any definition. And when we think about the historical perspective of this statement, this phrase, essentially free, first appeared in USP in 1946. So from 1946 until 2014, there was no definition around what we meant by essentially free. And so we think about essentially free today, it's a little bit different than we did back in actually 2010. And the last kind of key point that's outlined in this section, it talks about the inspection of visible particles can take place during the observation or investigation of other critical defects, such as cracks or defective containers and seals. But it was this idea that I talked about, what was essentially free, that led to the development of USP General Chapter 790. It became official in 1914, and as I mentioned, the first mention of essentially free was in the 1940s, so there was many years in between in which industry struggled to figure out what USP meant by this. The inspection conditions that are outlined in the chapter are harmonized with the EP and JP, and this goes back to the discussion that Rebecca was having earlier today. There are efforts within various pharmacopoeias to harmonize various aspects of the chapter, and the rationale is quite straightforward. If you know there's an expectation for visible particles in the US, in Europe, in Japan, if there could be some way to have a harmonized standard, it actually reduces the burden of the drug product manufacturer and having to do multiple tests looking at the exact same thing. The chapter really focuses in on extrinsic and intrinsic particles. Again, those particles that you normally will find within your manufacturing space, and those outside, such as dust and insect parts. The chapter does not deal with the topic of inherent particles. And as I discussed in the previous slide, when we think about inherent particles, these are things that are natural characteristics of the drug product. And so it's kind of hard to develop a compendial standard around something that may be extremely unique for a given drug product. And so the way we address inherent particles is that we say, hey, it's going to be addressed in an individual monograph, or it's going to be in the drug product's approved regulatory finding. But the big thing that gave, I guess, the new bit of information to 790 that really kind of pushed this topic forward within our industry was that we actually defined what essentially free was. And so essentially free is using acceptance sampling standard and an AQL of 0.65%. And this AQL value was actually developed through various PDA surveys looking at critical defects and the rate at which they were seeing failure. So it was a collaboration between USP and PDA and how we came up with this final definition of what essentially free actually is.
Desmond, with the visible testing, how is that done today?
It's done. It's a very manual technique where one has a white and dark background as shown in this slide here. You have an inspector who will swirl the sample, and then they'll look to see if they can see visible particles. So it's truly a manual form of visual inspection. But this 790 chapter is actually coupled with a much broader approach in looking at visible particles. The chapter in general, chapter 1, and the regulatory expectation is that 100% of all products have to be visually inspected. And usually this could be done through automated means, or manual. But again, we're starting to see a trend where it's being done more and more by automation. But to meet the expectation of essentially free, one has to do supplemental testing. Again, based on batch size, they test a certain number of products. And again, based on the AQL value, and meeting 0.65 determines whether or not a lot will actually meet the USP definition of essentially free. So this is today a very automated, a very manual approach. But again, the technology is out there today where you're seeing some really robust automated and semi-automated methods for looking at visible particles. So it'll be interesting to come back and have this discussion 5 years from now, or 10 years from now, and see where industry has gone in this respect.
Yeah, absolutely. Yesterday, I actually had a meeting with a customer who's building a new facility, and they are manufacturing sterile plastic bottles and containers for R&D, for media fills, for pharmaceutical testing. And they had a camera system along their line to visually check the bottles. And they're also thinking about using some AI technology to do some of the testing as well. So I can definitely see the emergence of automation and this new AI technology moving into this space, which will speed up the process and also eliminate human error, and take that out of the picture too. So you're getting good data on the process.
It's very interesting you bring up the topic of AI, because it's actually we just made a revision to our above 1,000 chapter where we kind of mentioned that this is a new frontier. I mean, there's a lot of data starting to be collected with, as you mentioned, on these automated systems, and so we just got to get to the point where we can kind of compare them to the manual method, and then the sky would be a limit as far as where the technology can be introduced and used.
Absolutely. And then also we can talk about the new Annex 1 standard that had been updated last year in August, I believe, and that talks about contamination control strategy, which harmonizes the whole facility and the collection of the data from different locations, different applications, from cleaning to particle counting to monitoring to microbial data, and it merges them all into a central system that will start to raise flags very quickly. If there's some problem along your process, you're able to quickly go and tune into to zoom into where that problem may be coming from.
Absolutely, it's this idea of in testing is where we are today. But again, as data starts to accumulate at different points within the manufacturing space, you can identify issues early on. Again, that's just with technology and the ability to generate so much data, that's where we're going. So let me continue. Some other key aspects related to 790, and this was kind of the discussion. In a perfect world, when you have a clear solution, dual visual inspection is quite straightforward, but we know that there are many products out there that we say are difficult to inspect. They may be in an amber container, or they may be a lyophilized product. So how do you, in these situations, determine whether or not there's visible particles? And so within the chapter, we add difficult to inspect instructions and how one should deal with them. Obviously, you cannot see the particles through the container, so you would have to withdraw it if it's a product in a dark container. If it's a lyophilized product, you have to reconstitute it. But again, one wouldn't expect that when you destroy the product in this manner that you're going to test thousands of samples. So again, this is more of a destructive test. So the number of test samples are reduced. And based on some of the data we collected, for most batch size, the number of product you would actually have to analyze in the destructive test manner way is around 20. And the acceptance would be no zero particles in those 20 that you actually tested. There was other discussions we have around what happens if particles are identified while a product is in distribution. And how does one go back and determine whether or not that lot is still essentially free? And so it's recommended testing of the retained samples to determine whether or not that lot is still essentially free. Again, looking at 20 samples or more. Again, if you have more within your facility, and again, accept that lot is essentially free if no particles are identified. The one thing we tried to stay away from, which I mentioned briefly when I was discussing the continuum between subvisible and visible, is that we do not define a specific particle size range for visible. Again, a lot of this is going to come down to the product, the training of your inspectors, your inspection setup. So USP didn't want to say it's 100 microns, and then someone said, well, we can go to 90 microns. So we just leave that open. But from most discussions we have within USP, we like to say that a visible particle starts at 100 microns. And the second piece is that we did not set an acceptance limit for inherent particles. And as I mentioned, this is very product specific. In USP, it wouldn't add value to create an acceptance criteria that wouldn't be utilized for most products anyway.
Yeah, and I think the onus should be on the end user. They understand their process, their product, and they could set their limits based on a risk assessment and good science and a baseline that they would have normally.
And so I agree with you, and I want to you keep that idea when we start talking about subvisible, because that's a pretty hot topic.
It sure is.
Yeah. In another chapter, a companion chapter that we have that kind of help give guidance on 790 is 1790. And again, people who aren't familiar with the USP architecture, chapters below 1,000, we tend to say are the mandatory in nature, because those are typically referenced in monograph, referenced in drug filings. And our chapters above 1,000 are meant to be guidance. What are current best practices related to a given topic? And so with the development of 790, we only thought it would make sense, as it was a hot topic, to develop something above 1,000 to kind of give end users more guidance or feedback beyond just the standard, which just tells you how to test and whether or not you pass or fail. And then 1790, again, it kind of gives you various general aspects that one should be aware about in their visual inspection program. Again, what are the key elements of an inspection process? What is the typical process flow for doing an inspection? What is the patient risk related to visible particles? And this is kind of a very hot topic. And I think I kind of alluded to that in my earlier slide. And when I say that not all particles are the same, I mean, some particles can be large but flexible versus particles that are large and rigid. Again, the risk profile can be quite different. And so it's the onus of the drug product manufacturers to understand the particles that are emanating from their systems and the potential risk related to those particles. The chapter talks about the elements of a good inspection process, life cycle and continuous improvement. Again, this idea of, as particles are being identified in your process, identifying what those particles are and putting them into your inspection set so that the inspector can look for these particles as they're doing their routine inspection. We talk about the different visible defect types. And as we were just mentioning a few minutes ago, Jason, various inspection technologies that are out there today, starting from manual all the way up to automated. So now I'm going to transition to subvisible particles. And this is a topic that has become near and dear to me. And I say this because in the last 5 or 6 years, there has been a lot of effort in this space for USP to either develop standards in the subvisible particle space or investigating or looking at some of our older standards to see if they need to be revised. So again, there's a lot of movement in this space today. obviously the chapter when we think about subvisible particles, the chapter that everyone knows about is 788 particulate matter and injections. And a little historical perspective on this chapter is that it was introduced into the USP in 1975. The chapter originally just focused on extrinsic particles and formatical, and it was really focused on those particles that at the time we felt would cause occlusion of capillaries. It was a simple test that really focused on large volume parenterals, and the only method at the time was the membrane microscopy method. In '84, again, in the evolution of a standard that we always see, we had the method, the membrane microscopy method for large volume parenterals, but we also introduced a new method for small volume parenterals, which was light extinction. And then in 1995 is when the light obscuration method became the preferred method in USP 788, which is method one, which still stands today. One should always start off with method one. If for some reason there's a product characteristics that leads to method one not being useful, such as viscosity, color, air bubbles, one would then move on to method two. And it was actually added to the chapter at the time because the ease of method control, the objectivity, and the efficiency. And what was going on in the background it was already being used by industry. From a historical perspective, many companies have had experience using light obscuration, and there were tons of products that had already been submitted for regulatory filings using light obscuration. So it became one of the official methods within the USP. We didn't eliminate the membrane method, but again, it became method two, and it became a tier testing with method one being chosen first. So in 2008 is when I think there was kind of a transformation in 788. And actually, I shouldn't say not a transformation, I guess a coalescing of ideas around subvisible particles for injectable drug particles. Because this was when the EP and the JP, along with the USP, decided to harmonize the chapter. Again, another topic that was brought up by Rebecca within the Pharmacopeia discussion group. And it's this idea that I mentioned previously that if there's going to be a requirement to do a specific quality test for a drug product in all jurisdictions, if the countries can figure out a way to harmonize it and agree upon it, it becomes a very useful tool or helpful tool for drug product manufacturers because they're not doing the same testing, slightly different depending on the jurisdiction. And this harmonized chapter is what we see today. Again, describes two compendial methods, light obscuration and membrane microscopy, includes default acceptance criteria based on historical experience. And Jason, this kind of circles back to your comment and question. For as long as I've been at USP, it has always been this discussion around whether or not the current limits within the USP are appropriate. Some will say they should be lower. Some say they should be not. And so we're having this kind of discussion around, should it be product specific, which goes back to your question. But again, from a compendial method standpoint, how do you develop a standard with acceptance criteria? So there's discussions we're having right now within the Pharmacopeia discussion group around acceptance criteria and what it actually means. So I think that would be something interesting to keep your eye on.
Absolutely, Desmond. And also as the technology in the light obscuration field, where we develop liquid particle counters, we're starting to go down smaller and smaller and smaller. And a lot of that's been driven from a different industry, which is the semiconductor, where the line widths on their chips now get down to below, around 5 nanometers. So getting higher resolution, obviously we need to move away from light obscuration to a light scattering technique in the liquid space. But as we move further and further and get more resolution, I think that will have an impact on your standards as well.
I agree, because as we start to look at things that were done, for example, some of these limits were back in the 1990s. The question is the more you see, what does that mean? And as you mentioned, analytical technology is getting better and better. And so what does this mean as we start to see more, as they say, stuff?
Yeah, absolutely. And then you have to define the probability of the impact of those size particles on the end product and the patient.
Yes, yes. So when we look at the chapter today, again, it requires the testing of 4 5 ml aliquots, which means you need around 25 ml of your sample to test. And so what was identified 2012 was there was one of our expert committee members who came to our meeting and said, well, guys, we're starting to have an issue. Well, 788 is a very good chapter. We know the historical perspective of it, but we're starting to see way more small volume parenterals and biologics out there that may have a fill volume of half of an ml or 1 ml. And so to be able to do 25 mLs of testing for a product that could be quite expensive or in development may be very difficult to get has been a struggle. And so this has opened up the discussion with the Pharmacopeia discussion group to look at revising the chapter to figure out if there's ways in which we could address this gap for small volume parenterals and biologics. Again, the ability to use smaller sample size volumes, additional sample preps, so that you're not going to that won't lead to the development of more particles when you're hoping to get less. So this is a lot of the discussion we're having right now as it relates to 788. And I would say keep your eyes open, because in the not so distant future, I would expect a republication of 788 based on these discussions around smaller sample size, more robust sample prep so that you don't create particles, to get stakeholders' feedback as we continue to have this discussion with the European Pharmacopeia as well as the Japanese Pharmacopeia. So 788 is definitely a harmonized chapter, everyone knows, but we also have other chapters that have been developed that kind of speak to the topic of subvisible particles. The one that I was most proud of was actually 787, because as I said, back in 2012, there was discussion around gaps with the current 788 as it relates to the biologics and small volume drug products. This is what led to the development of 787, which was particulate matter in the therapeutic protein injections. The idea was to modify that chapter to deal with small volume parenterals, as well as unique sample handling and other considerations for biologics. It was actually this chapter that really spurred the discussion with the Pharmacopeia discussion group. They saw the chapter, and the question is, wait, why don't we look at 788 again and revising it in such a way so that, again, we can take some of these lessons learned from 788 and add them into 788. And so at some point, one would expect, once we get to the end of the road with 788 and the revision of it, 787 would become obsolete. The one caveat I always say when it comes to harmonized chapters is that it is a very, I won't say torturous, but it's a very long process, because one could imagine every word, every sentence is analyzed not just by the experts within USP, but the experts within the European Pharmacopeia, as well as the experts within the Japanese Pharmacopeia. And again, those discussions can go back and forth vilely for years. But again, I think that we're getting close to the end of our process. So hopefully in the not so distant future, 788 will be revised, which will ultimately lead to the omission of 787. Another chapter that I definitely want to put on people's radar as it relates to subvisible particles is 771. It talks about the various product quality tests for ophthalmic drug products. It gives general guidelines for both visible and subvisible particles for ophthalmic drug products. And it gives a very unique perspective in that it outlines specific testing requirements depending on the route of administration. So depending on the route, maybe 788 testing and 790 may be required. There may be other situations where 789 may be required and 790, and there may be situations where only 790 may be required. So again, this chapter took a more granular look at ophthalmic drug products, looking at the various dosage forms and routes of administrations to give a more robust requirement versus something that's kind of broad today for some of their drug products. And then we have 789, which is the chapter that outlines the testing for ophthalmic solutions. It has just went through a revision and is actually going to be voted on. That revision, proposed revision, is going to be voted on by our expert committee in the next couple of months. And one, it would expect to see those out in the public space within the beginning of next year. And what the revision of the chapter really entails was making sure that the language that's in 789 is harmonized with the language in 771. And so one of the things that we did is the language that's used in 771 is interocular solutions. And so with the revision of 789, we made sure that that language was included in 789 so that when people are looking both of the chapters, it actually made sense and they didn't have to kind of figure out what we were trying to say when we meant ophthalmic solutions or ophthalmic injections in one chapter, and in another chapter we would say interocular solutions. Again, the test methods are similar to 788. The one big difference is that the specifications outlined in the chapter are way more restrictive.
Yeah, we've had some recalls recently from the FDA of eye solutions, and it is important to understand that the quality of these type of products is really important, because when you go through your eye membrane, you're getting right into your bloodstream.
Yes, and I have to say, when we've had discussions. I mean, and as Rebecca mentioned, any revision effort that we have that takes place within USP or a specific chapter, we always have government liaisons a part of the process. And again, those from the ophthalmic group, they're very conservative when it comes to many of the critical quality attributes that we talk about. And someone would say, like, why would the specifications be more strict for ophthalmics than injectable drug products? Well, I mean, they can give a host of reasons why are large particles important for the eye? Well, if there's too large, you can scratch the eye. So it was all these things that, again, as a scientist who maybe not an expert in that area, was able to kind of understand why ophthalmic solutions or interocular solutions are viewed with such conservative approach by the agency. So I have, this is my last slide, and so I want to kind of talk about two other chapters that, again, sometimes they slip through the crack because they're above 1,000 chapters. But as I mentioned previously, these above 1,000 chapters are meant to add more guidance. We have 1787, the measurement of subvisible particulate matter in therapeutic injections. And this chapter was really meant to kind of put out in this space all the current methods out today that are used to measure subvisible particles analysis. We give their strengths and their weaknesses. And we talk about how these various instrumentations can be used over the development life cycle of a drug product. It's one that, again, when I talked to people in the space about it, they didn't realize it was there. It's extremely valuable. Again, a chapter that became official 2015, and hopefully you can see this trend of new chapters in 2014, 2015, revised 2017, which goes back to my initial point that this has been an area of a lot of activity for USP and its expert committees. And then the last chapter, which was 1788. And this chapter was quite unique in that before USP harmonized a chapter in 2008, there used to be a very long 788 chapter with tons of stuff on how to standardize the instrument. That stuff was actually removed during harmonization and placed into 788. But what we decided to do with the revision in 2021 was to kind of break the chapter up into smaller pieces. So there's actually, you have 788, which is kind of an overview chapter of particulate matter. You have 1788.1, which talks about light obscuration. You have 1788.2, which talks about membrane microscopy. And then you have 1788.3, which talks about flow imaging. And the question people always ask is, wow, how did flow imaging get added to the chapter? Are you guys planning to put it into 788? Well, there was discussions at one time about including it in our below 1,000 chapters, but the difficulty or struggle we had, and Jason, I'm sure you can imagine, is it wasn't very easily to figure out what the specifications would be. And again, it's not apples to apples with light obscuration. And so we thought the best way to kind of introduce the technology into USP would be into 1788. And it's similar to as what was going on in 1995. Most companies are using the technology today, but again, its relevance within the USP or the compendium may take some time until more data has actually been developed. And so while one definitely knows 788, hopefully walk away with understanding that 787 and its role in the revision of 788, 771 and the product quality tests related to ophthalmic drug products, 789, but more importantly, that we do have 2 chapters above 1,000, 1787 and 1788. It's valuable guidance that can be used to help guide characterization of products and the development and qualification of methods. And with that, that ends my presentation on USP's world of particulate matter. And as I mentioned, it's a topic that we are actively engaged in, and there's other things that we're considering doing in the future. And to circle back to what Rebecca had said previously about USP cycle is about to end, and USP will start to look for new experts for the next revision cycle. So if there's anyone who is listening to this recording, please, if you're interested in being a part of the USP process in this area, please make sure you register to be a candidate for during our call for candidates. And with that, thank you very much. And I'm
Great, thanks, Desmond. That was a really good overview of the work that's involved, and it's also really good to connect with yourself and Rebecca, because we're really getting to the people who are inside of the USP organization who work on these standards and the framework of the organization, and in particular with your expertise in the field of particulate matter. It's nice to connect directly. So moving forward, we can always have a good reference of yourself as someone to contact and talk about the standards and the development in the standards in particular.
Yeah, and Jason, a point that I always like to say too is that a lot of times people feel like people within USP, our scientists like myself, can't be reached or there's not access to us, and that is so not true. We are definitely, what I like to feel like a part of my job is to talk about the standards and for people to better understand the standards. And I can't tell you the number of times I've had phone conversations or been a part of discussions where someone will illuminate maybe a shortcoming or maybe something that we had noticed in the standard or how the industry is evolving past the standard and we may need to look at it to revise it. So please, people should know that USP is kind of like an open door. They should feel comfortable with reaching out to me or Rebecca or someone within the organization if there's an interest in the topic or want to learn more about a topic.
Well, that's great to know, and I'm sure there will be people on this webinar that will be reaching out. Overall, I'm really impressed with the detail and the products and services that the USP organization has to offer. And I'd like to, it isn't the end of the webinar. I'm going to just move us on to, I've actually got an application I'd like to go over, which connects like the Lighthouse to USP and what we do a little bit in terms of, I'm going to focus on our liquid particle counters. And let me just get the screen shared with you again. So we did talk about light obscuration, and we have here in our presentation, let's get me out of the way a little bit, a technology that we've been using, like liquid particle counters. They're widely used in pharmaceutical applications and other applications like semiconductor industry. And like they're a lot of our customers in the pharma space, they use them for batch testing to ensure the quality, safety, efficacy, and the overall quality of the product and safety. And liquid particle counters, they're analytical instruments, and they measure the size and the number of particles in a liquid sample. They provide critical information about the presence of particles, including contaminants in the product, which can affect, as I said, the quality, efficacy, and safety of the product. And liquid particle counters are particularly important in the pharmaceutical applications, where particles can cause adverse effects to the product, to the patient. And I want to just highlight some of the FDA recalls over the years. Like we've had quite a few in terms of particulate matter in the products. And I'm just going to quickly show a few here that have popped up over the years. You can take a little bit of time to just read over them yourselves. I'm not going to read them out for you, but there's definitely been a lot of cases of FDA recalls on product, and especially when the quality of the product and the sample itself. A lot of these, you can see some of these products have missed that testing, where if you were to use the particle counter, the liquid particle counter, and we talked about the light obscuration, and basically how that works is that you got your beaker here with your sample, and that's quite a large beaker because we do actually sample a lot smaller volumes. But you have inside the sensor, you got a light source, laser we're using, and that laser is basically shining across a cell and the flow path. So you can see a tiny little tube here. This syringe here will pull down and let me, sorry about that. Click the wrong button. This syringe here will pull down and create a suction of this liquid here, which will go up through the sensor, and the flow path will take these particles through this area here, which is like, we call it the view volume. And that's where the particles will hit the laser. And what happens when you look over on let me just grab my little laser pointer. When you go over to this side, you have the particles passing through, and when a particle hits the laser, it blocks the light from this device called a photodetector. So normally when there's no particles going through, as in this diagram, you got a full flow of the laser to the photodetector. So we know the amplitude of the laser. So when we start putting particles through, that amplitude is going to drop. And then we're able to size and count the particles based on that signal dropping from the particle, which basically casts a shadow on the photodetector. And that's where we use the term light obscuration. Just to give a bit of a background on the technology. We do use these USP standards for the testing. We have these count cal particles, and typically we're looking at sizes of around 10 microns. And these particles come in a solution. There's this particular procedure we follow in the validation, and we follow 788. And we look at the counting accuracy by injecting the solution through. We know what the criteria of the solution is, and normally they come at a concentration of around 3,000 particles with plus or minus 10% tolerance. And once we run the sample through the particle counters, we have to verify that we're getting that count accuracy in that ballpark. And I just want to emphasize it's really important for us to be able to validate and prove to users the accuracy of the instruments that we're working with, because the criticality of the data that they're collecting on their product in order to release batches is so important. We have to have that level of traceability. So we have a metrology lab here ourselves, and we continually test even the standards that come in. We test them on with a higher resolution, and normally the light blockaging is around particle sizes of about 1 micron. When we start getting down to smaller sizes, we switch over to light scattering technique, which is a slightly different technique altogether, which uses the scattering effect of the light on the photodetector to give us the pulses of the amplitude changing from a particle light source, which connects to the photodetector. It basically transforms that into a digital signal that the sensor can read. For example, the amount of light scattered is proportional to the size of the particle. So then we're able to determine, oh, that pulse is a bigger particle, that pulse is a smaller particle. So it's an interesting technology. If you go back to the history of this type of technology, it was actually an Irishman, John Tyndall, who studied under Michael Faraday, who first came about this type of technology and understanding the relationship between particles and light, and the ability of later on, when we developed electronic equipment in the 60s and 70s, we were able to take that technology and automate it a bit more. So that's just a little bit of a background. I think we can move on to this next slide to wrap up. And I want to talk to, if I can hand it over to Rebecca or yourself, Desmond, just to talk about your vision for your global science and standards and where we're going into the future. I know we've touched on some of these things already.
Yeah, I could chime in quickly. I just see, again, at least in the space that I am in, is this idea of dealing with special products such as biologics and cell and gene therapy. I kind of hit upon the standards that we have right now as related to particulate matter and the challenges companies have had in the past to deal with them, especially as they're small volume. But even if you look at biologics and cell and gene therapy products and the special storage conditions, again, that's one of the things we saw as an avid of the COVID vaccine. So again, what are the special concerns around storage and distribution of products? And again, we hit upon the topic also of supply chain resiliency. Again, when we have a disruption in the supply chain, again, how does this impact drug product availability, which could impact drug shortages? So again, when I think about where we're going in this space, especially in the standard setting space, it's really in this area of looking at what standards we have today and how are they going to be adapted to deal with more and more products coming to the market that are biologic and cell and gene therapy in nature. And with that, I will see if Rebecca had anything else she wanted to add to that.
Desmond, I couldn't, I can't add any more to what you've already said. So thank you so much again for the opportunity, and I know I learned a lot today. So Jason, thank you for allowing us to speak on this. This has been a lot of fun for us.
It's been a pleasure, and we've learned a lot too, and a lot of our viewers as well. And we appreciate you coming on board and giving us a really good overview of USP. And the organization and what it does and the services. I'm particularly impressed with the ability to connect with the USP and ask questions about, standards and the knowledge space that you have there as well, and the training modules. So I'll definitely be moving over there soon and having a look. And we also have a knowledge center here at the Lighthouse. You go to our webpage, you can go there, and then actually this webinar will be available to view after we finish the webinar. I think around 24 hours, the webinar will be available on our knowledge center, and you can, if you haven't been able to join the webinar, you'll be able to connect and catch it on the recording. And again, thank you very much, guys. It's been a pleasure, and I look forward to working with you more in the future.
Thanks. I agree. Take care.
Thank you, Jason. Thank you so much.
Take care. All the best, everybody.